Good morning, good afternoon, and good evening, depending on where you are. I'm Zev Ronai, and I'm leading the Cancer Center here, and basic cancer NCI designated at the Stanford Burnham in La Jolla. And we are about to start our last seminar series for 2021, which is focused on precision medicine. The organizer of this year's seminar series have been Swasti Haracharyan, and she have put together an amazing seminar series that had duo presenters together with engagement of faculty and grad students and trainees that have the opportunity to meet with seminar speakers at the end of the presentations. Today's presentation will have a slight change. Unfortunately, Eitan Rupin was not able or will not be able to join us today. He will be rescheduled to make his presentation later on, probably early in 2022. Nick Wagle will present and will have the opportunity to use more of his time to make the presentation today, and I'm very much thankful for that. As a brief introduction, Nick have started at one point in my lab, and he may refer to that. That was, as he referred to, a blip, but have trained with some of the biggest linear data at the Brigham, and later on with Levi Garway at Harvard. He got his MD and did fellowship and residency at the Harvard and Brigham, and he's currently at the Dana-Farber. He's using translational research program in cancer genomic and precision medicine in order to characterize tumor samples from patients and to better understand molecular determinants of tumorigenesis. He is a director of Count Me In, a research initiative, a nonprofit organization that based at a Broad and Dana-Farber. And his CV is very impressive with respect to awards and nomination that he managed to achieve and obtain over the last actually relatively short time period, which is very, very, very impressive. So I can keep on with long introduction about productivity and work, but I rather we shift the gears to hear directly from Nick about his research and recent finding. All yours, Nick.
Thank you so much, Zev. And I'm just going to share my slides. Okay. Well, thanks for that introduction. Thanks to Swasti and Zev for inviting me to this series. It sounds like it's been a really great and exciting series, and I'm honored to be one of the speakers here. I will refer back to what Zev just mentioned. I was, my first lab experience when I was a college student in one summer happened to be in Zev's lab. That was my introduction and start to biomedical research, and it was the summer of, I think, 1996 or 1997, so a very long time ago. It was a place where I learned to do Western blots, I learned to do cell culture, and really my introduction to what ultimately became my career. And so it's particularly nice after many, many years to be reconnected with Zev and to be introduced by him for the seminar, and just wanted to acknowledge that that was such an important summer and moment for me. And as he mentioned, I've had the privilege of having really wonderful mentors along the way, beginning with Zev that summer. So thank you. Today, I'm going to talk about precision oncology and patient-partnered cancer research, focusing on some work that we've done in my lab in metastatic breast cancer, and then extending beyond that to understand how we might accelerate this vision of precision oncology. I'll start with a disclosure slide. And I'm going to start with just a case vignette to illustrate the challenges and opportunities in cancer precision medicine. So this was a patient who was diagnosed with metastatic melanoma at the age of 38. This picture was from now more than 10 years ago. And this gentleman had, you can see, metastatic disease. He had all of these subcutaneous deposits of melanoma all over his upper body. And if you saw the reverse picture, it's all over his back as well. And he, at the time, as you might know, there weren't a lot of great therapies for metastatic melanoma. We didn't have targeted therapies, we didn't have immune therapies. And this patient got a number of chemotherapies, some experimental work. He did even get some immunotherapy that were in early stages of investigation at that time, and nothing was working. He had a biopsy of one of his lesions, and that biopsy made its way to our lab. This patient was in another part of the country, and it made its way to our lab. This is when I was a first-year postdoctoral fellow in Levi Garraway's lab. We characterized that specimen, and we identified that it had a BRAF V600E mutation. And at the time, there were early clinical trials of RAF inhibitors. And so based on that finding, he received PLX 4032, which was the name at the trial of the drug vemurafenib that was in phase 1 studies at the time. And you can see he had this incredibly dramatic response. Everything just melted away within weeks. And unfortunately, this response, while wonderful, did not last. And a couple of months after being treated with vemurafenib, he developed a recurrence. And sadly, about 2 weeks after this third picture was taken, he died. He was, however, quite generous and was able to get a biopsy, which you can see here, the bandage of the healing lesion where he had the biopsy, and sent that tissue back to our lab for further characterization to help us understand resistance. And we were able to identify an additional mutation, a mutation in MEK, which is the gene just one downstream from BRAF, that was causing resistance to the RAF inhibitor. Unfortunately, although it was an important finding and helped us understand how we might design future trials and combination therapies, it wasn't fast enough of a discovery for him, and he wasn't able to benefit, but future patients certainly would or will. So I show this as a starting point for a couple of reasons. First, I think it highlights the opportunities in precision medicine. To be able to get this kind of a dramatic response, granted, this is one of the more dramatic responses one can see, but this is what we would like to try and achieve for all patients with advanced cancer. Find the vulnerability, have the right drug, and get these kinds of dramatic responses. The second reason I show it is it also shows some of the challenges, the challenge of resistance, which is in almost all cases, even when we have a great targeted therapy that works, resistance invariably develops. And so to better understand how this can happen, ideally to preempt or overcome resistance in all patients going forward, just as we had learned in this patient. The third reason I show it is just that this was my introduction to this field. This was the first patient that I studied as a postdoctoral fellow. It's the first paper I published as a postdoctoral fellow, and really got me started on thinking about this idea that a single patient's donation, a single patient's tumor sample could help us learn so much, and helped to, over time, think about how can we take this and amplify it and scale it. If we can learn so much from one patient, what can we learn from many patients? And I will come back to that idea in a little bit. So with that in mind, the work that we do focuses on translational genomics and precision cancer medicine. And we really have, in my group, 4 major goals. One is to understand the biology of cancer and mechanisms of metastasis. Second is to identify predictors of metastatic disease. The third is to elucidate mechanisms of drug response and resistance. And the fourth is to use what we learn in 1, 2, and 3 to discover new therapies and therapeutic strategies. And we've taken the approach of trying to address these questions, just as in that patient with melanoma, initially through the study of clinically annotated tumor specimens from patients with metastatic breast cancer and more recently metastatic cancers. And so today I'm going to have 2 parts to my talk. I've extended it a little bit given the time. The first part, I'm going to talk about the work we've done in the lab focused on resistant ER-positive metastatic breast cancer. And then in the second part, I'm going to talk about how we've started to expand this approach through a program called Count Me In, which allows us to partner with patients even more to accelerate this type of research. So metastatic breast cancer is the most common cancer in women in the US, with more than 250,000 new diagnoses per year. About a third of patients will develop metastatic breast cancer, which is breast cancer that is spread beyond the tissues of the breast. And there's an estimated 155,000 women and men living with metastatic breast cancer in the US, and over 40,000 people in the US die from metastatic breast cancer each year, which accounts for 14% of all cancer deaths among women in the US. And although we have a number of treatments and treatments are improving and we continue to get new treatments approved for metastatic breast cancer each year, there's currently no cure for metastatic breast cancer. And this is largely due to the problem of drug resistance. So I'm going to focus on the most common subtype of metastatic breast cancer and breast cancer called estrogen receptor positive breast cancer. And in this cancer, it typically starts out sensitive to drugs that target the estrogen receptor. The estrogen receptor is a dependency in this cancer, and we've known for many decades that targeting the estrogen receptor is a way to successfully target this cancer. And so now we use a combination of hormonal therapies, including tamoxifen or selective estrogen receptor modulators, aromatase inhibitors, which decrease the amount of circulating estrogen, and fulvestrant or selective estrogen receptor degraders, which degrade and antagonize the estrogen receptor itself. And more recently, we have now combined hormonal therapy with targeted combinations, including CDK4/6 inhibitors, and the mTOR inhibitor everolimus, or the PI3 kinase inhibitor alpelisib. But in essentially all cases in the metastatic setting, while tumors often start out sensitive to therapy, they ultimately, just as that patient with melanoma did, develop resistance to therapy. And the questions that we've been asking are, how does this resistance occur? And can we develop therapeutic strategies that can overcome or prevent this resistance? And so the approaches we've used to study this type of resistance in breast cancer include a combination of focus on patient samples and laboratory-based approaches, using both laboratory-based approaches in models to identify potential mechanisms of resistance and genomic and molecular characterization of tumor specimens from patients who develop therapeutic resistance, and looking at the intersection of these two sets of findings to understand what's most clinically relevant. And so the main driver of our data to date in metastatic breast cancer has been the study that we launched at Dana-Farber Cancer Institute in our Center for Cancer Precision Medicine, now going back to 2015. And this is a study in which patients who come to Dana-Farber who have resistant metastatic breast cancer are asked if they would be willing to participate in this biopsy study, whereby they agree to a metastatic re-biopsy, where we get a biopsy of their tumor. Often these are metastatic and resistant tumors, as well as a sample of their blood. And we use those to study resistance, evolution, and tumor heterogeneity. And so we obtain a specimen, we profile them for clinical decision-making, we look at the pathology, we look at the receptors, we look at genes that are altered that might be influence clinical trial enrollment, and then we use the remaining tissue to do a number of research-focused studies, including whole exome sequencing, transcriptome sequencing, single-cell RNA sequencing, and spatial proteomics. And over time, we collect serial plasma cell-free DNA from these patients for additional whole exome sequencing. The important thing here is that since these are all our patients at Dana-Farber, these are linked to detailed clinical information, including their demographics, diagnostic information, pathology, treatment response, and radiology. And so over about 4 years, we collected 509 biopsies from 446 patients with metastatic breast cancer. And over time, we have been characterizing these tumor samples. And so we have now this number's increased 593 tumor specimens and 1,300 blood specimens. And we've completed whole exome sequencing on about 300 tumor specimens along with RNA sequencing. And we've started to increase the numbers of samples in which we've completed single cell sequencing and spatial analysis. And so today I'm going to focus on the results from our exome sequencing from tumor and blood. We've presented some of the other work at recent meetings, and we'll be presenting more at the San Antonio Breast Cancer Symposium later this month. But for today, I'm going to focus on the exome sequencing studies. So the first thing that we looked at when we looked at the exome sequencing from these patients with resistant metastatic ER-positive breast cancer is, does the landscape of genomic alterations look different than what we already knew about primary treatment-naive breast cancer? And what we see here is these are genomic alterations found in 236 metastatic biopsies from 198 patients. And we can see that there is a large overlap with primary treatment-naive breast cancer, for example, from the TCGA study, which is not unexpected because these are breast cancer driver genes and one expects to find them both in primary and metastatic disease. But we also found that there are a number of additional genes that were not significantly mutated in primary breast cancer that we do see mutated in this cohort of metastatic samples, and they are highlighted here, and I'll get into them in more detail in a few slides. And this has now also been seen by a number of other groups that have done genomic sequencing of metastatic breast cancer, namely that there are a number of genes that are mutated in the resistant metastatic setting that we don't see mutated in the primary treatment naive setting. So this raises a question: are these cohorts different because tumors that are destined to develop metastatic disease have these mutations and that makes them more aggressive and more likely to be metastatic? Or are these mutations that are acquired and selected for and enriched over time? And to look at that, we have the option, because in these patients we follow with biopsies serially, we can look at pairs of samples or series of samples where we look at a baseline tumor biopsy, either primary diagnosis or before a particular endocrine treatment, and we look at the biopsy after treatment and we compare the two. And so analyzing these pairs of metastatic biopsies or cell-free DNA in patients with resistance might help us identify what's acquired and what are the mechanisms of resistance, which then can help us think about targets for combination therapies. And so here we look at the mutations on the left that are present in the resistant sample or the second sample that are not present in the first sample. And so here's an example. We look at the cancer cell fraction, which is the estimated number of cancer cells in a given biopsy that have a particular mutation, and compare that to the cancer cell fraction in the MET. And for many of the genes, we see that they're shared. They're in this upper right corner where they're present in 100% of both samples. There might be some that are lost or are present in 100% of the primary, but not the MET. But our real interest is in these ones that are present in 100% of the MET, but not in the primary, and these are the acquired mutations. We've recently developed an additional way of looking at amplifications that way, where we correct for copy number and ploidy, and then we look for the copy number changes above changes in ploidy between the pre and the post, and we can look for gain of acquisition of amplifications. And so looking at those two, we took pairs of samples where we have two samples per patient, with more than 120 days of ER-directed therapy between time points. And we had 60 patients that fit this criteria, where we had pairs of samples with 4 months of ER-directed therapy in between. And we compared what was present in the post-treatment specimen with what was present in the pre-treatment specimen, very similar to the melanoma patient, but now across 60 patients who received endocrine therapy. And we saw a number of interesting things. The first is, in about a quarter of the patients, we see acquisition of these estrogen receptor ligand binding domain mutations. This has been well known for a number of years that patients with resistance to endocrine therapy have ligand binding mutations in the estrogen receptor, which makes them estrogen independent. Here, we confirmed that. We confirmed that they're present in about 25% of patients. And we also confirmed what was well assumed, but here we can show it with pairs of samples, that these are all in fact acquired. This has been known in the field, but what hasn't been known in the field is what causes resistance in the remaining 75% of patients with resistance to endocrine therapy. And here we were excited to see that we found a number of other important patterns. In another mutually exclusive group, we found HER2 activating mutations. So these are mutations in the HER2 receptor in mostly in the kinase domain that cause the HER2 to be constitutively active. Now, these mutations are different than what we typically look for in breast cancer, which is HER2 amplification. These are, for the most part, HER2 non-amplified breast cancers that have occult and acquired mutations in HER2 that causes them to be active. And so this was a good candidate for another mechanism of resistance to endocrine therapy. And then in a third group of patients, also largely mutually exclusive, we saw acquired alterations in the FGFR pathway, including FGFR1 acquired alterations, FGFR2 acquired alterations, and FGF3 acquired alterations. Acquired is everything that's in red or brown in this graph. And so in a series of several publications now, which are all out, we went through the HER2 and the FGFR pathway mutations and started to show that these were sufficient to cause resistance to endocrine therapy. I mentioned that we want to cross what we see in tumor samples with what we see in laboratory studies. And so one thing that gave us confidence that we were looking at the right genes is a parallel study we did where we did a large-scale open reading frame gain-of-function screen to look for mechanisms of resistance to estrogen receptor degraders. So here we took ER-positive breast cancer cells, we infected them with a genome-scale library containing about 12,000 different genes. We treated them with fulvestrant or a different selective estrogen receptor, GDC810, over time, and we looked to see which genes, when present in the cell line, were sufficient to cause resistance to the estrogen receptor degrader. And when we looked at this, we found a number of genes that cause resistance to both fulvestrant as well as GDC810. And when we started to look at which genes these were, a lot of them, looking at gene set enrichment analysis, fell into these four categories: FGFR signaling, ER+B signaling, insulin receptor signaling, and MAP kinase signaling. And so when we look back at this list of genes that we found enriched and acquired in the resistant sample, we see our ER+B2 and FGFR1 that also come out of the screens. And the reason I selected several of these other genes is that these also fit with the genes that come out of the screen, including IGF-1R amplifications, as well as a number of genes that are in the MAP kinase pathway. And so taken together, we found that 63% of these patients, 38 out of 60, have an acquired alteration in one of these genes that in our screen, and then subsequently in validation experiments that we did, are sufficient to cause resistance to endocrine therapy. So what are we learning from these studies of tumor samples and laboratory studies? We now know that there are at least these three classes of resistance mutations to endocrine therapy: ER mutations and fusions, which have been extensively published on over the last 10 years or so. Now, HER2 activating mutations, as well as FGFR pathway amplifications or activating mutations. And all three examples we see are acquired in resistant ER-positive metastatic breast cancer. Two, importantly, detectable by profiling metastatic biopsies or cell-free DNA. And three, potentially targetable with drug combinations. Each of these now has a therapy that can be combined with endocrine therapy to overcome resistance. And I don't have time to talk about it now, but we've shown in vitro that this works. And now there's also patient data that's starting to show that this approach works. The other thing that was striking to us is that when we started looking at these different candidate mechanisms, as well as the other ones that we saw in the MAP kinase pathway, that it seemed like these might be all converging on a similar pathway. And so we introduced these alterations in FGFR1, 2, in HER2, and in ESR1 we engineered them into ER-positive breast cancer cells. We grew them up in the presence or absence of various endocrine therapies or other drugs. And then we collected RNA from these cells. And we did RNA sequencing across all of these. There were a total of 909 different drug combination and genomic perturbations. And we did RNA-seq. And here is what it looks like when we just look at the parental lines with the various genomic alterations in the absence of any drug. And what you see here, interestingly, is that the FGFR1 and 2 and the HER2 mutants start to cluster together, the RNA-seq results, and separately from HER2 wild type, the controls, or ESR1. When we looked at differentially expressed genes in these different cell lines, we see some similarities. In the cells that have HER2 mutations, we see, not surprisingly, genes that are in the ER+B1/2 pathway are up. And we also see, somewhat more surprisingly, that RAS MAP kinase pathway genes are enriched, and that the estrogen receptor-driven genes, the canonical ER genes, are mixed. They are not all up, some are down, whereas an alternative ER syndrome driven by growth factors is all enriched. And so what we see here is that when you have these HER2 mutations, they activate the MAP kinase pathway, and they transition the ER from the canonical syndrome to a different set of genes driven largely by this growth factor. And we see a very similar thing in the FGFR1 and 2 activation. We see RTK growth factor receptor genes up, and we see RAS MAP kinase signaling genes up. And when we target both of these sets of cells with MAP kinase pathway inhibitors, like MEK inhibitors, we also see reversal of the resistance mechanism, suggesting that there is a common node that's activated here. And so when we combine all of these, this was the work done by Ophir Cohen in my lab, looking at what genes are common to both the HER2 activation signature and the FGFR1/2 activation signature, we start to see a common cell state, which he has named RTK-ACT, or RTK activation, which seems to be an emerging paradigm in resistance to endocrine therapy here through the activation of multiple different RTKs, which are all converging and activating the MAP kinase pathway. We've now started to corroborate this idea in multiple other studies. And so here is another study that we published last year focused on resistance to CDK4/6 inhibitors. Here we looked at a series of biopsies from patients who received CDK4/6 inhibitors plus endocrine therapy. And we looked at samples that were resistant versus samples that were sensitive. And we saw a number of genetic alterations that were enriched in the resistant samples versus the sensitive samples. And those, we selected several of them for further study, and those are shown here. RB1 loss, AKT1 activation, KRAS or HRAS activation, Aurora kinase amplification, cyclin E2 amplification, and again, FGFR2 and ERB2 activation, as well as some patients who lose the estrogen receptor altogether. And interestingly, these 8 mechanisms were present in 66% of the resistant tumors profiled. But what's important is that many of these mechanisms converge on this MAP kinase pathway that we saw in the FGFR and the ERB2 samples as well. And so, when we tested these, when we put these in vitro and tested them for whether they could cause resistance to endocrine and CDK4/6 inhibitors, they did, and were reversible by targeting these mechanisms or targeting pathways downstream of these mechanisms. I want to say one last thing about what we're seeing in resistance in ER-positive breast cancer, which is this idea of ER loss. And so in about 10 to 20% of samples, these tumors lose the estrogen receptor altogether. And it's not really well understood either how they lose the estrogen receptor or what they do to substitute for estrogen receptor as a driver. And here I'm showing 4 examples of patients who get some series of drugs targeting the estrogen receptor or chemotherapy, and over time, they lose the estrogen receptor. They go from being ER positive on the left to being ER positive on the right. And we've started to characterize these patients genomically, and we're starting to see some interesting patterns where they are developing additional alterations, either in transcription factors related to ER, like FOXA1 or GATA3 or AR, as well as more typical oncogene mutations like KRAS or AKT activating mutations, which may together substitute for ER loss. And this is an early work that we're hoping to develop more. And so just to summarize what we're learning about resistance to endocrine therapy in ER-positive breast cancer, we see that after tumors develop resistance to ER and CDK4/6 inhibitor, we seem to be seeing 3 classes of resistance. The first are secondary alterations in the target, where ER mutations or fusions or amplifications develop that cause resistance to the endocrine arm. And others have recently seen CDK6 alterations, as well as there's some emerging reports of CDK4 alterations that might be causing resistance to the CDK4/6 inhibitor. Here, I focused a lot on the compensatory or bypass mechanisms, where we see RTK activation, MAP kinase pathway activation, or cell cycle alterations, or in some cases, PI3 kinase pathway alterations, which together seem to cause cross-resistance to both ER and CDK4/6 inhibitors. And then lastly, there's this emerging idea of pathway indifference, where we see either loss of ER expression or, even with the presence of ER expression, transition to another intrinsic subtype, which has been seen in other tumor types like prostate cancer and lung cancer and melanoma, where the tumors really change their cell state and require an altogether different approach. And that's an emerging, I think, an important mechanism of resistance in this tumor type. And so what does this mean for precision medicine? So we're learning a number of things about these tumors, namely that the metastatic setting looks different and has a number of these alterations that aren't present initially, and that many of these are targetable. And so this means that if we are profiling the metastatic samples, we might see many of these alterations shown here. They occur, most of them at very low rates, but all of them are targetable in some way. HER2, RB, AKT1, FGFR2, mismatch repair deficiency. And so this might allow us to profile resistant specimens and then assign various trials based on what we're seeing in the metastatic setting. In the CDK4/6 inhibitor patients specifically, we now know enough about the different resistance mechanisms that we might think about constructing a basket trial focused on the different alterations that occur, because each one of these alterations, which probably account for more than 50% of the resistance mechanisms in these patients, have some associated therapy that is likely to have some effect in these patients, or at least we think will have some effect in these patients. I also want to highlight that you could think about targeting each individual resistance mechanism, but as I said, many of them seem to be activating common downstream nodes, whether those are Aurora kinase or MAP kinase or AKT and mTOR or CDK cyclin E2. And so we've recently been thinking about doing these more nodal type trials where we target, for example, the MAP kinase pathway in either many patients or all patients to try and overcome resistance that's caused by many other effectors. Over time, I think there's the opportunity to use a combination strategy that might target all nodes or drivers. And because this might be toxic, maybe you have to do these through a rapid cycling where we cycle through every different node. And ultimately, it would be wonderful to try and move this up front to prevent resistance. But of course, a lot of work needs to be done to be able to get there. So what are we learning about resistant ER-positive metastatic breast cancer? Well, I hope I've convinced you that one, metastatic resistant breast cancer looks different from primary breast cancer. Two, that's at least in large part because these tumors evolve as they develop metastatic disease and as they develop treatment resistance. Three, because of this, it's really important that we do metastatic tumor biopsies and blood biopsies because they have both research implications, but also clinical implications. Four, there are several key nodes of endocrine resistance, even though there's probably a long tail of resistance mutations that we could find. They seem to start to converge on a couple of key pathways, including the MAP kinase pathway. Five, DNA sequencing does not tell the whole story, and we're now very interested in focusing on other aspects, RNA sequencing, protein analysis to understand the full landscape of resistance mechanisms. And then six, this does have, right now, clinical significant implications because we have drugs that target these resistance mechanisms, and we are starting to develop clinical trials to see if these improve the outcomes for patients. So a vision then for cancer precision medicine would be to use this type of information for refined stratification of all patients using diverse molecular features, to deploy rational combinations tailored to individuals, to continue longitudinal sampling of tumor tissues and cells, and to do, when possible, real-time monitoring of response and resistance and tumor adaptation with real-time optimization of these therapies. We still have a lot of work to do to get to here, but I think we can see in the field of cancer genomics and cancer precision medicine a move towards this because we're learning a lot, and I think we will be able to get closer to this vision in the coming years. So in the second part, I want to talk about how we might expand and accelerate this vision. And so how can we accelerate these types of research advances? Things that we need are increased scale, because we learn a lot from one patient. We may learn a lot from a large study that's done in a single institution like Dana-Farber Cancer Institute. But to really make progress, we need lots of patients. We want to learn the same amount of information for many thousands or tens of thousands of patients. We need comprehensive genomic and molecular characterization because we only know what we look at, and the more we look at, the more we can learn. We need this to be coupled with detailed clinical annotation. I think one thing we learned from the TCGA approach is that we learn a lot about the biology of tumors, but without clinical annotation, it's hard to apply them to specific clinical scenarios like resistance. We need a study with modern treatment regimens. And so the studies that exist are starting to get outdated. And as we get new and more clinical drugs approved, some of the what we've learned is going to evolve and it's going to change. And finally, we need these studies to include underrepresented and historically excluded patients, because our understanding of these cancers needs to be representative of everyone who gets these cancers. And to date, we've not been very successful at including everyone. And so one way to do this is through collaboration and data sharing, and that clearly accelerates discoveries. And there's a number of examples, some of which are shown here, of institutions and organizations that are doing an incredible job of making their data publicly available and sharing data and really making it a public good for people to learn from. And so that's one way that we can accelerate these types of discoveries. One issue though, and what I've spent the last 8 years or so thinking about, is that the vast majority of cancer patients are not treated at those large research institutions. By some measures, 85% of cancer patients in the US are treated in community settings. They get their care, those samples and those records go into their local hospitals or local doctor's offices, but they aren't used for research studies. And so the fact is that in the US, most patient samples and data have not been readily available for study, for the simple fact that most patients in the US have never been asked if they would be willing to share their samples and their records for research. And so back in 2013, 2014, we started to think about this problem. We were seeing the kind of data that we were generating in our local patients at Dana-Farber and wondering if we could expand this to include this large number of patients who aren't able to participate and aren't offered the opportunity to participate in similar types of research studies. And the idea was that technology, social media, and cultural changes, which now have more organized advocacy groups and engaged patients and advocates, now provide an opportunity to engage cancer patients directly and partner with them in this type of research. And so this is where the idea for Count Me In came. The idea that every patient's experience with cancer should be counted, that every patient should have the opportunity to share their medical information, their specimens, and their voices with all researchers, and that every community should have the opportunity to participate and contribute to research so that our research advances reflect every community and every person. And so with that in mind, we started this initiative called Count Me In. And you can see our website here at joincountmein.org. It says every patient's experience with cancer should be counted. And this is a joint nonprofit research initiative between the Broad Institute, the Dana-Farber, the Emerson Collective, and formerly the Biden Cancer Initiative, which is no longer in existence. And the simple goal of Count Me In is to enable cancer patients anywhere to share their information and samples with researchers everywhere. We started this out as a pairing to the work that we were doing in metastatic breast cancer at Dana-Farber, focused on metastatic breast cancer patients. And so in 2014 and 2015, we spent a long time talking to patients around the country, understanding how they would want to participate in a program like this, what would make it appealing for them, what would make it easy for them. And with the combined efforts of many patients and advocates and advocacy groups, we launched this project called the Metastatic Breast Cancer Project, found at mbcproject.org. And you can see the website here says, help transform our understanding of metastatic breast cancer. It says your tumor and your medical records could unlock discoveries, and ask patients to become part of the research movement and have a direct impact on the future, and walks people through how they can participate. And if patients are interested in participating, they go to that button at the top right that says Count Me In, and then a box pops up. It asks for their name, their email address. They check a box that says, I have been diagnosed with metastatic breast cancer, and I would be interested in potentially sharing my information and my biospecimens and my experiences. So after spending about a year talking to patients and building this project and building really the whole concept of this project and the Count Me In initiative, we launched this in October of 2015. And over the last 6 years, more than 6,000 women and men with metastatic breast cancer from all 50 states and US territories plus Canada have joined. And this is a large study in metastatic breast cancer. As far as I know, I think it's the largest prospective study of metastatic breast cancer, certainly that collects clinical data and genomic data that exists right now. And so once people sign up, we've developed a process and approach to engage them. So we send them an online consent form that asks for their permission for us to obtain and analyze their medical records, their tumor specimens, their saliva, and their blood remotely. We collect longitudinal patient-reported data through surveys. We ship them a saliva kit and a blood collection kit, which they can provide, and then they drop it back in the mail, and that comes to us. They tell us where they got their care, and then we contact their doctor's offices. We request and receive medical records, and then we read those medical records and find out where their tumor specimens are stored. And then we request portions of their tumor tissue from institutions across the US and Canada. Then we read the medical records and we abstract the data from the medical records as well as from their patient-reported data. We perform the same kind of detailed genomic characterization that I discussed in the first part of my talk on tumor and saliva and plasma-derived cell-free DNA. And then we combine all this data. We analyze, we interpret it, both somatic and germline. And then importantly, part of the pact with patients who are participating is that we share all of this de-identified data that we either acquire or generate from the project on a regular basis. So in real time, pre-publication without restrictions via public or accessible databases like the CBio portal and dbGaP and the NCI Genomic Data Commons. Importantly, patients, patient advocates, and patient advocacy groups have been involved from well before day one in conceiving, designing, implementing, testing, and refining this project. And in the breast cancer project, for example, there are more than 40 different advocacy groups who are part of this effort, which in breast cancer is, it's wonderful to see this number of advocacy groups working together for a single cause. This is the parallel chart to what I showed for the Dana-Farber study. Here we have 6,000 patients registered, 3,400 who've signed consents. And the biggest bottleneck for us is actually now going through and getting all the data and getting all the samples. And so we've collected 2,000 saliva samples, 1,000 blood samples, 600 tumor samples, and we've been making progress in generating the genomic data from across a number of these tumor samples. Importantly, as a proof of concept, we looked at where patients were getting their care. And this is a chart, a figure that shows that patients are getting their care from all over the US and Canada. Of the first 2,589 patients we analyzed, only 6% reported receiving their care at NCI designated cancer centers, whereas 1,174 institutions, 70% of them that were listed were reported by only one patient. And so we do represent some of the largest cancer centers here, shown in the middle, but most of these patients are coming from a place where they're the only people participating. And so this is the proof of concept that this allows us to be synergistic with the type of work that happens at the big cancer centers by enrolling patients who otherwise might not have a chance to participate. And so this is what we can learn from patient-reported data. We can survey them, ask them about their age at their primary diagnosis, their years between the diagnosis of metastatic disease and registration, or years between their initial diagnosis and metastatic disease. And we start to be able to build these cohorts. And the cohorts are sizable. So 1,100 patients diagnosed before the age of 40 allows us to study young women with breast cancer, which is a particular area of interest of mine. We have 1,200 patients with newly diagnosed metastatic breast cancer. So think about following these patients longitudinally. We have 165 patients who've been living with metastatic disease for 10 years or more, which is much outside the expected life expectancy for someone once diagnosed with metastatic disease. And so what can we learn from these extraordinary patients who've lived for a long time? And similarly, thinking about the time between initial diagnosis and metastatic disease, we have over 1,000 patients diagnosed with de novo metastatic disease, which allows us to study metastasis that is not confounded with treatment resistance. And similarly, we can study large numbers of patients with late recurrence more than 6 years after initial diagnosis. We have treatment information on all these patients, and so we can look at various treatment classes versus the amount of time that patients are on them. And we can look for these outliers at the top of patients who have been on particular therapies for extraordinarily unexpectedly long periods of time and understand, is there something about their tumors that we could learn? And the types of studies that we might be able to do by combining tumor analysis, germline analysis, medical record, clinical data, images and pathology, and patient-reported data is really large. And here are just some examples of the types of studies that we're interested in either doing or having other people download the data to do. And so to that point, all of this data is publicly available, and we've been sharing it on a regular basis since we started this project. You can see all the different places where the data is stored on our webpage or by going to one of these sites. And there is a fair amount of use of the data so far. So right now, the MBC project, we've not yet published a paper. We have a presentation and a preprint that hopefully will come out in the next month or so after San Antonio Breast Cancer Symposium. We've not published a paper, and yet this data has been used by more than 30 peer-reviewed papers using the data. There are others in preparation and under review. And we noticed a significant increase in data inquiries and downloads, particularly over the past couple of years, coincident with the COVID-19 pandemic. And so our hope and what we think is that this data is being used as it should, as intended as a public good. So in the last part of the talk, I want to expand beyond breast cancer. We were interested in the model that studying breast cancer provided in Count Me In. And the question we asked then was, can we do this in rare cancers? And so we launched a second project in 2017 focused on a much rarer cancer called angiosarcoma. Angiosarcoma is a cancer that occurs in the endothelial cells. It can occur all over the body. It's extremely rare. There are about 300 cases in the US every year. And we are sorely in need of new treatments. There isn't a great standard of care. And for patients with aggressive or recurrent or metastatic disease, we really don't have a very good standard of care at all. And that it's quite aggressive and lethal in those patients. And so we launched a second project focused on angiosarcoma, modeled after the Metastatic Breast Cancer Project. And you can see a similar website here. And over the first few years, more than 600 women and men with angiosarcoma joined the project. And so that's a substantial number of people who've been diagnosed with angiosarcoma over that same period of time. When we started looking at the tumor samples that we acquired in these, we didn't take looking at very many before we started to notice patterns. And that turns out to be one of the real benefits of this approach, is the ability to study rare tumors. And so you can see here on the left, we studied 36 tumor samples with whole exome sequencing from these patients. But because we had a fairly large number relative to what had been studied, we were able to group these based on the place in the body where the angiosarcoma arose. And so you can see here, some of these in red are cutaneous radiation-associated angiosarcomas, usually after breast irradiation. Some of them are primary breast angiosarcomas. Some of them in orange, for example, are angiosarcomas of the head, neck, face, and scalp, and so on. And by looking at the genomics crossed with the site, we started to notice patterns that were site-specific. So if you look in this top right, you can see that the tumor mutational burden that we notice is different depending on the site in the body where the angiosarcoma arises. And in particular, these tumors that arise in the head, neck, face, and scalp, angiosarcomas arise in the head, neck, face, and scalp, have a significantly higher tumor mutational burden. And when we looked at the genomic signatures associated with these, they were all UV damage signatures, suggesting that there was some UV damage associated in the pathogenesis of this head, neck, face, and scalp angiosarcoma, which makes sense since these are in sun-exposed regions, leading to a high mutational burden. Now, this is very reminiscent of melanoma, which has a similar genomic signature and high mutational burden. And so the question we asked is, if this is similar to melanoma, might angiosarcomas of the head, neck, face, and scalp respond similarly to treatments that work well in melanoma, namely immune checkpoint inhibitors? Now, fortunately, there was no immune checkpoint inhibitors at the time approved in angiosarcomas, but we had treatment records from all of these patients. And so we looked at the treatment records to see if any of these patients happened to have received immune checkpoint inhibitors off-label because their doctors decided to give it a shot. And we found 6 patients in the cohort that had received immune checkpoint inhibitors at some point in their care. 3 of them had head, neck, face, and scalp angiosarcoma, and 3 of them did not. The 3 that did not, none of the 3 responded to immune checkpoint inhibitors. Of the 2 that did have immune checkpoint inhibitors, of the 3 that did, 1 of them came off immediately because of a reaction and never got the immune checkpoint inhibitors. And the other 2 both had extraordinary responses. Both were patients that had widely metastatic disease, not responsive to therapy. Both got pembrolizumab. Both, after less than a year of pembrolizumab, came off treatment, and now multiple years later have no evidence of disease and are seemingly cured. So highly sensitive to this immune checkpoint inhibitor. When we went and sequenced these tumors, we did find, in fact, that both had very high mutational burden and had a UV signature that was dominant. And this going back before the FDA approval of pembrolizumab, led the sarcoma community before our paper was published, really just when we first observed this pattern, to develop clinical trials of immune checkpoint inhibitors for patients with head, neck, face, and scalp angiosarcoma. And so the time from discovery to clinical development was incredibly short, and this was in part due to this patient-partnered approach. So we've now launched projects in more cancers, in prostate cancer, in gastric and esophageal cancer, in brain tumors, including pediatric brain tumors, in osteosarcoma, which is a pediatric bone tumor, and colorectal cancer. And we've started to see an increase in enrollment across all of these cancers across the United States. Back in August, just a few months ago, we decided to expand further, and we opened up Count Me In to anyone who's ever been diagnosed with cancer at any time, any stage. And so this is now, Count Me In is truly Count Me In, open to anyone who's ever been diagnosed with cancer. We were able to partner with Stand Up to Cancer. So Reese Witherspoon back in the August telecast announced Count Me In, and we got a nice bump in enrollment since then. And with Count Me In, we have this nice PSA that's running on television and radio, trying to get patients with cancer to sign up. And in the first couple months since this launch of all cancers, we've had about 1,000 new patients sign up and over 100 new cancers represented. So we're really hopeful this model will allow us to study many cancers, particularly rare and understudied cancers. I think opening up to all cancers is not sufficient because, as I mentioned in the very beginning, it's important for us to be inclusive and particularly to include patients that are underrepresented or historically excluded from these types of studies. And so we have a particular effort trying to focus on community engagement and cancer equity, which has a number of different arms of how we're trying to be more inclusive and be more representative. And I'll highlight two of them. One is a project that we just launched within the last week or so called Amplifying Black Voices Across Cancer, which aims to lift and amplify the voices and lived experiences of Black patients with cancer and share their stories. And so we now have this website, www.blackcancervoices.org, which is in soft launch right now. We'll launch more in the coming days, which features many of these stories. And the hope is to try and build a community in whom we can talk about research and try and make sure that we are more inclusive. And then all of our materials have been fully translated into Spanish. And so we're working with members, Spanish-speaking advocates and patients to try and make sure that we're inclusive of Spanish-speaking patients as well. So I'm going to close the second part of that talk there, highlighting the power of patient-partnered research. The reason I think this approach is really important and powerful is that it's patient-centered. It includes patients who are treated in community settings. It enables more diversity and inclusion in research, which is an opportunity for all communities to participate. It gives us the potential to enroll very large numbers of patients, which allows the study of questions that require large numbers plus the study of rare subgroups. Everyone is prospectively consented and able to be recontacted, which is a differentiator between many of the studies that look retroactively at patients. We have serial and resistant samples with genome-scale multi-omic data, and this is really a convergence of clinical, genomic, and patient-reported data, all linked to one another and linked to other data sets. And the data is publicly shared as it's generated with no restrictions. But perhaps most importantly, and the way to summarize this is a true patient-researcher partnership. And you can see here, this is over the first year or so of the studies, where patients were going into social media and sharing pictures of themselves with their saliva kits or with their blood kits, or they were sending in kits decorated with stickers or notes or thank yous, or they'd even come and visit back before the pandemic when that was possible, and meet the researchers and meet the lab and explore and see what we were doing, and we would meet them. And it's really been a fulfilling thing to do, not just because I think it accelerates cancer genomics and precision medicine, but because the way in which it does that through this patient partnership is incredibly important. So I'm going to close there. I'll just summarize, and then I'm happy to open it up for discussion and questions. One, profiling tumor biopsies is a powerful approach to studying resistance mechanisms. Experimental approaches using screens, models, and other modalities are necessary to interpret results of tumor analysis. Two, multiple genomic mechanisms of clinical resistance have been identified in resistant tumors, and many of these are acquired, detectable by tumor or ctDNA profiling, and targetable with existing or emerging therapies. Number three, there's likely to be a long tail of specific resistance mechanisms, but it may be possible to group these into a smaller number of targetable pathways or nodes. And transcriptional analysis may help to identify these nodes in tumors with both known and unknown resistance mechanisms. And Dr. Rupan, who was going to present today, is an expert on this, and hopefully, his work is going to shed important light on this. Number 4, approaches to target resistance mechanisms include basket trials, combinations in resistant tumors, or upfront combinations to prevent resistance. Number 5, further profiling of metastatic tumor biopsies and cell-free DNA, coupled with open data sharing, ideally, is needed to study resistance mechanisms, to identify biomarkers of response and resistance, and to develop new treatments and treatment strategies. And finally, number 6, partnering with patients can accelerate inclusive cancer genomics and precision medicine research, particularly the study of questions that require large numbers, studies of rare subgroups, and studies that include underrepresented or historically excluded participants. So thanks very much for your attention. Thanks again for the invitation. I want to thank my lab, as well as the entire Count Me In team shown here, as well as all of our collaborators and our funders. Thank you very much.
Thank you so much, Nick. I love that we ended the seminar series for this year on this note, because I really feel like this is such an evolving field in how we're doing cancer research and how we're treating patients. Obviously, lots of questions from the audience about the Count Me In stuff, but I'd like to talk about the biology a little bit before we move into the more, I know, extremely fascinating stuff that you're doing with Count Me In. So we got a couple of questions about this, so I'll ask you those as easy ones, and then we'll talk about the actual origin of why we're asking these questions. So, one of the questions was that in the metastatic ER-positive breast cancer setting, you know that you get HER2 mutations in the recurrent setting, right? So what are the issues with just treating these women with HER2 inhibitors as a first-line therapy instead of waiting for recurrence?
Yeah. Great question. I think that's an important paradigm, which is when we learn about a resistance mechanism, we're already a little bit late if we observe the resistance mechanism and then treat it. And I think you could make that argument for everything we do. All resistance, by the time we see it, we're late, and we're better off combining therapies up front. I think to get to that point takes some time. So we are still at the stage where we're observing and learning the resistance mechanism. Then we need to show formally that targeting that resistance mechanism with a HER2 inhibitor, for example, is effective and safe, particularly if we're talking about combinations where the safety is an actual question. And then we need to do the trials to show when you do it up front, you're better off than the current standard of care. And that cycle takes a long period of time. There's one more part of that cycle, which is then you move it to the adjuvant setting and you prevent more people from recurring. That is definitely the ultimate goal, but we are still early in that cycle to be able to do that. But I completely agree with the people asking the question that is where we want to head.
So, and it almost as a follow-up to that, for example, with the ER positive, we're seeing all these MAPK resistance mechanisms, right? Mm-hmm. But we know that even if we treat with MAPK inhibitors, we're going to have further resistance mechanisms. So is there a way to address that? Can we start off with more aggressive combinations, or are we still lacking those biomarkers that will tell us who's going to need more aggressive treatment and who's going to respond?
Yeah, super question. I think the worry about the whole study of resistance in general is that we are chasing our tails or playing a game of whack-a-mole, depending what analogy you want to use, which is you target MAP kinase and then you lose ER, or you target MAP kinase and then you develop a PI3 kinase mutation, and then it's never good enough. Well, I think a few things. One, if it's better than current, it still may be worth it, right? If you're going to extend the lives and the quality of these patients while we're still trying to figure out a cure, we should be doing so. I think there's a lot short of a cure that is still worth doing. And so if targeting MAP kinase pathway is better than current, it's worth it. The second is that as these tumors evolve, there are a couple of different possibilities. One possibility is that every time you target something, it evolves to something even scarier, and then you're left with this really aggressive tumor that we don't have anything to do. But another possibility is that we are blocking off different escape routes, and we are cornering the tumor into a certain state, which maybe has some really important vulnerability that we can target. An example might be immunotherapy, right? Immunotherapy doesn't work great in ER-positive tumors right now, but it's possible that if we've targeted enough of the escape mechanisms and it gets to some certain state, that may be more amenable to immunotherapy, and we have some evidence that that may be true. The third is there might be a better paradigm of targeting these resistance mechanisms. And so I agree, combinations, being more aggressive upfront, or like really creative paradigms like observing when the resistance mutation occurs over the course of therapy and then adding something instead of switching. Those are all things that are being talked about and I think would be really good. The problem is that it's really hard to change the drug development paradigm and the clinical trial paradigm. And so if we want to do stuff like that, we're going to have to figure out creative ways to do this clinically.
Yeah, and sort of to get to the origin stories of these questions, right? I mean, really where it's coming from is there something about some ER-positive breast cancers or any type of cancer really that makes them more able to activate these resistance mechanisms? So in other words, are there just two types of breast cancer, right? Ones that can evolve and ones that are less able to evolve. And so that's why we're having such an issue with drug resistance, because these have something intrinsic in them that helps them find alternate pathways more quickly or more rapidly before the drug can have an effect.
Totally, super question. I think the answer is probably yes. And I think there's some examples that there are that a mechanism of resistance, or maybe a higher order mechanism of resistance, is like you're saying, the ability to evolve, whether that is genomic instability or some other lack of repair that allows tumors to develop various alterations, I think is possible. And we probably see that, mutations with tumors with P53 mutations tend to be resistant in general, maybe not because of the P53 mutations, but because of what that signifies. Hypermutated tumors tend to be, have lots of different mutations. They just have more opportunity to develop resistance. So I think that is part of it. I think another part of your question, which is fascinating to me and I don't understand, is why is there a dominant resistance mechanism, right? So why do some tumors have ESR1 mutations and some tumors have HER2, and why do those largely look mutually exclusive? It could be a sampling thing, and it could be that when you look deep enough, they all have everything. But it does seem to be that most tumors have something dominant. And even when you start to see convergent mechanisms, they tend to converge in the same group, like they have multiple ESR1 mutations, but you don't see any HER2 mutations. So I think that's a really important area for someone to figure out, which is there something intrinsic to a tumor which predestines what type of resistance it's going to get? And I think that probably yes, but I don't know what that is.
And sort of related to that, I think, which is because I work on ER-positive breast cancer, I think about this a lot, which is some of that predisposition to your major resistance pathway because of the very unique evolutionary history of, say, something like ER-positive breast cancer versus something like triple-negative breast cancer, because ER-positive breast cancer is so focused on that G1S cell cycle phase because it's responsive to that mitogenic stimulus from the hormone, right? And so it feels like even in your studies, for example, the major resistance mechanisms are how to overcome that mitogenic stimulus at G1. So either you do the CDKs and the cell cycle stuff that regulates the G1S progression, or you go upstream and you start activating alternate mitogenic stimuli like HER2 or FGFR or those other receptors that can compensate for the loss of the endocrine stimulus, right? Which could explain why in triple negative, for example, if you survive, you're good. Whereas for the ER-positive breast cancers, you have this really long recurrence window because you have this ability to sort of circumvent this mitogenic stimulus issue.
Yeah, great point. I think that's totally right. And I think the lineage specificity is really important here. And one thing I talked about MAP kinase, and I had it in the one figure as separate from, say, CDK2 cyclin E, but it's very possible that what MAP kinase is doing is ultimately driving growth through CDK2, right? So maybe everything converges on CDK2 ultimately, right? You target ER and CDK4, and CDK2 becomes the driver. And maybe it's actually much simpler than that, and we just have to target CDK2. I think that's possible.
I really like that. That's my pet hypothesis. So I'm glad you agree that that's possible. Related to that, one of the questions Peter raised, he had to leave for thesis defense, but I think it's also interesting in terms of the specificity of resistance mechanisms, is why is it that even though we have this dependence on G1 progression, we're not really seeing that many mutations in the P16RB pathway, right? We're not seeing mutations in P16 or cyclin D1. We have the CDK4 specific effects. So why do you think that is? What is it about ER-positive breast cancer that's doing that, or is it across cancers?
Yeah. So
I feel like that's a tricky one.
It's a tricky one. So let's start with cyclin D, for example. So we know cyclin D1 is frequently amplified in ER-positive breast cancers, and we know that maybe cyclin D1 actually is a marker of responsiveness to CDK4/6 inhibitors. RB is required for CDK4/6 inhibitors to work, and so loss is a mechanism by which you can essentially make those irrelevant, although in vitro loss of RB is still maintains sensitivity to endocrine therapy, to targeting the ER. So I think the RB loss is specific to the CDK4/6 inhibitors, not to targeting the whole pathway. Whereas if you did something similar in cyclin D, I think that's too much of a dependency, right? So cyclin D is essential for these tumor cells to grow. And so if there was a cyclin D loss, it would probably be more akin to tumors not being ER positive anymore. And it may be that it's easier for tumors to lose the ER than it is to lose cyclin D or P16, for example. But it's a good question. And it may also be that we haven't found the mechanisms by which these happen, because we've only looked at certain types of alterations.
Yeah, no, I totally agree. So then let's switch gears to talk a little bit about the Count Me In project and how fascinating it is. So there were some questions about, when you do the Count Me In, do the local physicians or oncologists also get to be a part of the research, or is it all just funneled directly to you?
Yeah, it's more the latter, but it's something we've been thinking about a lot. So the idea here was to engage directly with patients, and they're the ones driving participation by signing up online. It was not intended to leave out local physicians, because of course local physicians could be part of it, and in many cases are, for two reasons. One, either patients say, I heard about this Count Me In project, and they go to their doctor and they talk to them and they say, should I participate? And the physicians or local caregivers, figure out whether this is worthwhile or what they think about it. The second is, in some cases, people who have heard about Count Me In, or who we know, or who take care of cancer patients in other parts of the country, whether it's at academic centers or not academic centers, have just really taken it upon themselves to steer their patients towards this. We have not figured out a good way both to engage clinicians around this, or to, I don't want to say reward, but basically to include clinicians who are taking it upon themselves to include their patients. And it's something that I've been thinking about, and we want to do more. We have some ideas, but it's definitely an area that we could develop more. Right now, it's largely been focused on patients.
And so do you then talk to the patients directly to give them feedback on what you learn, or does that just go through the local clinician or oncologist?
Neither. Well, no, not neither. So when we don't, at least in most of the projects that exist right now, we don't return individual results to patients, right? This is done in a research setting. So it's essentially a donation of tumor samples that we study. And what we share with patients is we share aggregate information. So we share what we're learning, we share our progress, we share status. If we're presenting a poster at San Antonio next week or this week, that we will, we'll send that out in advance to patients to view, that kind of thing. And similarly, that information is public, so physicians can look at it. We do have two new projects, one focused on osteosarcoma, one focused on leiomyosarcoma, supported by the National Cancer Institute. And as part of those, we are piloting return of individual results to patients. And there, we're going to return them directly to patients, but also have the option to get it to their doctors as well. And so we'll see how that goes.
So actually related to that, and this is my favorite question asked in the Q&A, so maybe we'll end on that one, is, do you envision that this Count Me In project could be a way to actually do real-time therapeutic studies, like clinical trials? Like, can you envision a way this could be modified to say, hey, we've learned all this, and we think these patients that we just got the tumor samples from should go on HER2 inhibitors now, because we think they'd benefit the most from that, and talk to the local clinician or something? Or is that just too sci-fi?
No, not sci-fi at all. That's definitely the vision. The vision is for this to become bidirectional. So right now it's a lot of patients sharing and then that being used for research. So it's like, it's circular, but it's not bidirectional. the goal is, what can we do that will directly impact patients and patient care? And so I would love it if we can develop a way that we can either return information in a timely fashion to patients that might help with their care, or what we're learning about patients can be directed to someone who's running a trial that can then contact that patient. I think the former is a little hard because we're not a diagnostic testing company that's doing this for the explicit purpose. So there's a time lag. And so people should not be signing up for Count Me In because they're looking for an answer in 2 weeks. There's lots of other opportunities that people can do that. Our process takes a lot longer, but it could be that one of the really powerful things that we have is we have this network of really engaged patients who are looking to help researchers, but also I'm sure would be very happy to help themselves. And if we could say, we take this engaged network and we somehow connect that with an engaged network of clinical trial investigators or pharmaceutical companies looking for patients, I would be very happy to have that matchmaking ability.
Yeah, that would be amazing. I really think this was, the perfect talk to end this year with. Thank you so much for joining us and giving us all these really great things to think about in terms of cancer research. I really appreciate it.
Yeah, well, thanks for having me. This has been great. And I look forward to spending the rest of the afternoon meeting folks. And yes. I wish it could be in person, but I'm glad we could do it over Zoom.
Agreed. So I'm going to ask the trainees who are staying on for the roundtable session to stay on, and everyone else is welcome to leave. Thank you for joining us. Join us again next year.
Thank you.